focusing on one of the most important biological processes we observe in our laboratory experiment, the mitosis. In plants, mitosis is especially active in regions where growth occurs rapidly, such as the root tips. This is why in our experiment, we use the onion root tips. Since onions grows fast and contain many actively dividing cells. This process is crucial not only in plants but in almost living organisms because it allows cells to divide, multiply, and replace old or damaged cells. We observed mitosis in an onion root tip. At first, we prepared our slide and placed it under the microscope, expecting to see clear stages of cell division. But when we looked through the eyepiece, the cells were not visible at all. Everything looked blurry and too thick. We realized that our first slide wasn't properly prepared, the root tip wasn't thin enough, and the stain didn't spread evenly.
I'll begin with the first stage of mitosis, prophase. During prophase, the cell undergoes major preparation step before dividing. In our onion root tip slide, we observe that the chromatin, which is normally spread out in the nucleus, begin to condense into visible chromosomes. These appear thick and dark when viewed under the high power object. Another important change is that the nuclear membrane gradually breaks down. This allows the chromosome to later move freely within the cell. At the same time, spindle fibers start forming from the centrioles to and plants. Spindle formation occurs without centrioles. Based on our microscope observation, prophase cells were easier to locate because their chromosomes were clumped together instead of being spread out.
After prophase, the cell enters the second stage, metaphase. Metaphase is one of the most recognizable stages of mitosis because the chromosomes are arranged neatly at the center of the cell. This general region is called the metaphase plate. When we viewed the onion root tip slide, it was very clear that the chromosomes had lined up in a straight, organized formation.
The cell proceeds to anaphase, which is one of the most dramatic stages of mitosis. In anaphase, the sister chromatids separate. These chromatids are exact copies of each chromosomes, and during anaphase, they are pulled toward opposite poles of the cell by shortening spindle fibers. Under the microscope, we observe anaphase cell where the chromosomes look like they were being dragged apart. They form distinct V-shaped structure, which is a characteristic sign of chromatids being separated. This stage is crucial because it ensures that each new daughter cell will receive a full set of chromosomes. Without proper separation, cell might end up being with missing extra chromosomes, which can cause abnormalities. In our onion root tip slide, anaphase was easy to identify because of the visible pulling motion of the chromosomes away from the center.
The final stage of mitosis is telophase. During telophase, the chromatids have completely moved to opposite ends of the cell. At this point, the chromosomes begin to decondense, returning to their usual thread-like structure. A new nuclear membrane forms around each group of chromosomes, creating two nuclei inside one cell. In our microscope observation, telophase cells were noticeable because they showed two forming nuclei. which indicated the cell was almost ready to split.